we hope will be a really exciting new strategy to treat, provide treatments for patients with really advanced degeneration. So why do we really focus on photoreceptors, replacing the photoreceptor cell? Well, that's because loss of photoreceptors is the main cause of blindness, both for inherited disease and an AMD. And we think that retinal repair by photoreceptor transplantation might provide a generic treatment for all kinds of disorders. And we also think that it might be feasible. It might be one of the most feasible types of repair in the central nervous system, because photoreceptors only need to make one very short connection to connect to the rest of the visual pathway, and that's quite dissimilar to other neurons that make very long connections and make very many of them. So I think it's going to be much harder to repair other types of neurological disorders. But having said that, it's still a huge challenge, a huge scientific and technical challenge. But what we need to do is to break it down into its component parts. And we have to try, and that's what we and others are doing around the world, is to make progress in each of a number of steps. And the major requirements, we think, are identification of appropriate donor cells, and secondly, finding ways to transplant those cells successfully, have them integrated and making functional connections in the retina. And both of those are huge challenges that require extensive research. And I'm going to give, it's going to be a lot of concepts today and a lot of data, and I'm going to try and give you an overview, because I think it's important to realise just how much science is required to get this into the clinic. It's not a new area of research. Scientists have been trying to repair the retina, transplant photoreceptor cells for over 100 years. And so it has a long history. But it's not been something that there's been much success, because it's very difficult to transplant photoreceptors and have them migrate and make functional connections. But in 2006, we, in my lab, had I think a major breakthrough where we demonstrated that it is possible to transplant photoreceptor cells effectively, provided the photoreceptor cells are at a particular stage of development. And this was a study in mice. All that I'm going to talk about now it's all done in mice. So you know, we have to eventually translate that, but we're trying to establish some principles, some framework for this type of therapy. And what we showed, and this is just a summary, just to give you some background, because this study was the basis of all our current program. What we did was we took photoreceptor cells from donor mouse retina, and the photoreceptor cells were labelled with a green fluorescent protein so we could identify them when they were transplanted. And we made dissociated cell preparations from these donor retina, taken from mice of different ages, and injected them subretinally into adult mice. And what we found was that if we injected cells that were almost fully differentiated adult photoreceptor cells, but not fully differentiated, they hadn't elaborated all the outer segments, these cells were able to migrate and integrate into the retina, and they were pretty much indistinguishable, morphologically indistinguishable from the host photoreceptor cells. And this was the first time that anyone had ever transplanted photoreceptor cells. And what's really important about this finding, I think, is that it this knowledge can be used to generate the appropriate cells for transplantation from embryonic stem cells, or induced pluripotent stem cells, or even adult cells. We know the stage the photoreceptor cell has to be at. And in this study, we also transplanted into animal models of degeneration, and we could show that we could restore some kind of function following transplantation, that they appeared to be making connections within the retina that we could measure using electrophysiological techniques. And we could even show that there was a pupil reflex, that these the animal models that we were transplanting in had a very poor pupillary constriction to light. And the particular model we used was the rhodopsin knockout mouse that has no functional rods. And we were transplanting these new rod photoreceptor cells, and we saw that the pupils were then able to constrict under dim light. Now, we're injecting very many cells. We're injecting 200,000, 400,000 rod precursor cells. And at this point, only about 1,000 cells would integrate. So we're having to use very sensitive techniques to pick up function. So we had some evidence that the cells were connecting, but what we really wanted to demonstrate was whether the integrated cells were making functional connections in such a way that we could restore vision. And we certainly at this point couldn't do that. So we spent another 5 years trying to answer this question. If we transplant new photoreceptor cells, can we restore vision? And in order to do this, we optimized our transplantation technique, and we also changed the model we're investigating. And we're now using a mouse model of stationary night blindness. It has a gene defect in GNAP1, which is rod transducin. And that means that the mice have no functioning rods, but the rods are present, and there's a very slow degeneration. And what we found is we could, with our new techniques of transplantation, we could get very many more photoreceptor cells integrated into both normal mice and these adult GNAP1 knockout mice. So we take a suspension of cells, inject 200,000, and now we're getting up to 40,000 integrated cells. And that was a huge breakthrough because we now could test function in a very different way. And on this very busy slide, I'm not going to go over this slide. This is just a summary of a paper we published earlier this year in Nature, in which we look to see whether we can improve vision in these treated animals. And this is the paper. It was published in May. And what we could do, we could identify many more cells, so we could look in great detail. We could see that we restored normal physiology, that we have now transplanted cells that contain the alpha transducin. Remember, the alpha transducin is missing in the endogenous cells. And not only that, the alpha transducin behaves in a physiologically correct manner. It moves in between the different photoreceptor compartments in light and in night and day. So the transplanted photoreceptor cells are behaving in a physiologically correct manner. We could also look at the synaptic connections of these newly integrated photoreceptor cells, which are marked with this green fluorescent protein. And we can see that the synapses contain all the appropriate synaptic proteins like dystrophin, bassoon, ribeye. But more importantly, what we could now do is look in detail using electron microscopy to see whether the synapses really look normal. And this is an image taken from a textbook, an old textbook, showing the structure of a rod ribbon synapse that makes a connection between the horizontal cells There's ribbon synapse there, and the bipolar cells. So if we now look at transplanted cells, these are all pseudo-coloured. These are the horizontal cell, these are the bipolar cell. You can see here's a transplanted photoreceptor. There's the ribbon synapse. So it's looking pretty much indistinguishable from a normal rod photoreceptor cell. And that was a very important proof. So the first time we could really show it's making an appropriate connection. And I think more importantly, or equally importantly, we could actually then look at the electrophysiological functions of the photoreceptor cells in vivo. And this was using a technique called patch clamping, and this was carried out by King-Wei Yao at Johns Hopkins. And he could show, looking at individual photoreceptor cells, transplanted cells we can identify by the GFP, that they have the same kind of electrophysiological properties as the normal rod photoreceptors. But I think what was really starting to become very exciting is when we saw that not only were they making functional connections in the retina, but that the signals from these transplanted cells were actually going to the brain, and we carried out an experiment to look at the activation of the visual cortex in treated mice. And if we assess visual cortex activation in dim light conditions in treated and untreated mice, we see a very different pattern of activity. So in dim light, in the visual cortex, there's no activation of the visual cortex. But when we look in treated animals, we start to see that the visual cortex is active, and that's the first indication now these transplanted cells are making functional connections to the brain. Doesn't mean the mice can see, but it's the start of the possibility. It suggests the possibility they may be able to do so. But in order to see whether they might see, we have to carry out behavioural tests. And one test we carried out was so-called optokinetic testing, where we're looking for a reflex response to a moving grating. So mice do this without any training. If there's a moving grating, they will track their heads. You can watch them move their heads in the direction of the rotating grating. And we can use this to measure both